a little bit about very powerful methods of detection of different analytical objects. It can be large toxic molecule, it can be virus, it can be bacteria, whatever. Terminology in this area is mostly biochemical and medical, so it's very different from what we see in chemistry. And mechanism of many of these processes is not established well. What's important that it works. So the object we want to determine or to detect is named antigen in biochemistry and medicine. So when antigen binds immune cell, special cell that works for protecting human body or say animal body or cell from some objects, it results in activating this immune cell. So immune cells make more antibodies. Antibodies are molecules that bind this antigen. Again, antigen can be virus, can be cell, can be simply a molecule. Now, what's next in the animal world? There are different molecules named macrophages that eat antigen and together with antibodies. So the whole goal is selective destruction of antigen. So this approach can be utilized for analytical purposes. Antibodies, molecules, this is just an example, can be very complex. So this is usually a very complex protein group that is partly selective towards molecule or group of molecules. First analytical usage was radioimmunoassay related to radiochemistry, chemistry of radioactive species. It was historically the first. There is still some limited usage of it, but the principle is the same for all the methods. This approach is connected with the name of Rosalind Zussman, the lady who was born in Bronx, New York, and lived all her life and died in Bronx, New York. She had Nobel Prize in 1977 for this method. Her field actually was physics, not biochemistry, but since that she switched to biochem and applied her deep knowledge of physics for her work. So the idea is we have known amount of radioactively labeled antigen. So we know what we are looking for. We label it radioactively and then put it in a beaker or, small vessel. Then we add fixed amount of specific antibody. Next, we add anti-immunoglobulin antibodies that precipitate all these antibodies we already added. So at the bottom of our vessel we have precipitate that is radioactive because of our radioactively labeled antigen. Radioactivity of this precipitate after we separate it can be measured. Now, For unknown sample, we add exactly the same amount of radioactively labeled antigen and add exactly the same amount of antibody. Now there is competition between labeled and unlabeled. So some of labeled antigen is left in solution. So after precipitation, we have less radioactivity of precipitate. We have some radioactive from our addition, and we have some non-radioactive in precipitate from our sample. Then we measure this radioactivity. It is always smaller than starting. Next, what we can do, we just make calibration curve. If we use just usual scale, so concentration of antigen and radioactivity, will have a curve which is not very convenient. So we usually use log scale. In this case, we have usual straight line, and we can use it as our calibration curve. In here, cold means non-radioactive. It's language of radiochemistry. Now, this approach, of course, requires to work with radioactive species, and we mostly do not like this. So the most common approach these days is enzyme link immunosorbent assay, or ELISA. What is this? In case of viruses, our antigen, we have our virus sample absorbed by a surface. The antibody with enzyme conjugate attached to antigen like that, and excess of antibody is washed out. Now you have enzyme on the surface, and then you run enzymatic reaction, changing color or doing something else. More enzyme, brighter is the color, which means more virus, more antibodies on the surface, more enzyme. You build calibration which will be linear for your antibody, as a result linear for your virus or any other antigen. Now, it's not always easy to sorb your antigen on the surface, so you may want to put another antibody, antibody 1, that will selectively or maybe unselectively bound your analyte. So you have polymer support, you have analyte bound to this support. This can be protein, can be other molecule. And next you add second antibody that is labeled with enzyme. In this case, again, number of your enzymes is exactly the same as number of your analyte. As a result, enzyme works producing colored product, which is visible. Alternatively, it produces fluorescent product, which fluoresces. These will give you even better sensitivity. In modern devices, we mostly use fluorescence unless we are using visual detection. Instead of enzyme, you can bind something very strongly fluorescent. In this case, after that, you can see fluorescence of bound antibody directly, but fluorescence we can detect with enormous sensitivity. It's one more scheme. You have captured antibody, you put target protein on it, you are using detection antibody. And next, you attach enzyme to this, and next you run your enzymatic reaction. Just practical example. Horseradish peroxidase, very famous reagent. When you have running this product, aldehyde with amine makes bounded surface, and you have your reaction. As a result, you have colored plates showing different amount of your antigen. It can be competitive reaction or it can be linear. Very sensitive option would be chemiluminescent reaction. What is this? Luminol and several other compounds reacts with hydrogen peroxide in presence of enzyme to make such aminophthalic acid and emit light. In this case, you can count single photons coming from your sample, and your detection methods would be very sensitive. So again, you just put in appropriate catalyst here, and you have chemiluminescent version of ELISA. All these methods have plenty of modifications and are widely used in toxicology and biochemistry and in medicine.